You know that little pill you might take?
Yeah.
Its journey from like a scientist's lab bench
Yeah.
all the way to your medicine cabinet. It's this epic saga, really,
Yeah.
full of twists and turns.
Absolutely.
And if you're anything like us, you want the full picture. But maybe without getting totally lost in the weeds, you probably wondered, how does that actually work?
Right.
So today we're diving deep. You've got this fascinating bunch of material that really lays out the whole pharmaceutical drug development process.
Exactly. We're going to trace that entire path. I mean, from the very, very first spark of an idea for a treatment all the way through to the crucial monitoring that keeps happening even after a drug is out there for patients.
Yeah, the info you shared gives us a really comprehensive look at, well, the whole thing.
It really does.
And our mission for this deep dive, it's basically to pull out the most important nuggets of knowledge, those aha moments, the bits that really show how new medicines get made.
And highlight some surprising facts along the way.
Yeah, and hopefully provide some clarity without totally overwhelming you. We want you to get the why behind each step, not just the what's.
Makes sense.
Okay, so let's jump in. Where does this incredibly complex multi-stage process even begin?
Well, it all kicks off with understanding the disease itself, right down at the molecular level. We call this stage target identification.
Target identification, okay.
Yeah, think of researchers like molecular detectives. They're trying to pinpoint a very specific biological molecule, maybe a protein that's gone haywire, or an enzyme working too much or too little.
Or maybe a gene.
Exactly, or a receptor sending bad signals, or even a specific gene that's playing a key role in the disease.
So it's like finding that one specific domino.
Yeah.
The one that, if you could just nudge it right, could stop the whole disease cascade.
That's a perfect analogy, and finding that domino involves some incredibly sophisticated tools and a ton of information.
What kind of tools?
We're talking genomic studies mapping out our entire genetic code. Proteomics studies, looking at all the proteins buzzing around in our cells, and then powerful bioinformatics to make sense of all this incredibly complex data.
Wow.
Yeah, and it's amazing how just comparing the genomes of people with and without a disease has uncovered totally new, sometimes really unexpected molecular players.
Huh.
And what's really changed the game is that the range of potential targets has just exploded. It's not just about traditional small molecule drugs anymore.
Yeah, you hear about other things now.
Exactly. We're seeing amazing progress with biologics, with gene therapies, RNA-based treatments. It's opened up a whole new universe of ways to potentially intervene.
Okay, so they've zeroed in on a molecule, looks like a key suspect in the disease. What happens then? Do they just like throw chemicals at it?
Not quite yet.
Yeah.
The next step is absolutely critical. Target validation.
Validation.
Meaning confirming that messing with this target will actually do something useful therapeutically. And crucially, that it's likely to be safe. Just because a molecule is linked to a disease doesn't mean changing its activity is the right answer or won't cause other problems.
Okay, that makes sense. So how do they validate it? Seems like there could still be a lot of guesswork.
There's definitely still uncertainty, which is exactly why they use several different techniques to build confidence.
Like?
Well, they might use something called RNA interference, RNAi, or maybe even CRISPR gene editing.
Oh, yeah, CRISPR.
Right. They use those in-cell cultures or maybe animal models to essentially switch off or tweak the target gene and see what happens. Does it actually reduce the disease signs in the model?
Okay.
And they also do pharmacological validation. That means using specific chemicals, inhibitors, or activators that interact with the target protein to see if that produces the desired effect. Really nailing this validation step early on is just so important. It stops companies pouring huge amounts of time and money into a target that, ultimately isn't going to work out.
Fail fast, fail cheap kind of thing.
Exactly. Or at least validate thoroughly before committing massive resources.
Okay, so validated target in hand. Now, does the image, I have labs full of scientists carefully mixing chemicals, come into play? Finding the first drug candidates.
That image isn't entirely wrong, but the process is way more high-tech and faster now. This stage is called hit identification. It's about finding those initial molecules, the hits, that interact with the validated target.
Hit identification.
And a real workhorse here is high-throughput screening, or HTS.
High throughput. Sounds impressive.
It is. Imagine these highly automated robotic systems. They can test thousands, even millions of different chemical compounds against the target simultaneously.
Millions? Seriously?
Oh, yeah. Using these tiny miniaturized tests in the lab, it lets them quickly flag molecules that bind to the target or change its activity.
Wow. That sounds like searching for a needle in a haystack. or maybe a specific grain of sand on a beach.
It definitely can feel that way, and that's where virtual screening becomes so valuable, using powerful computers.
In silico, right?
Exactly, in silico. These techniques let researchers screen enormous digital libraries of compounds. Some might not even exist physically yet.
How does that work?
Well, there's structure-based drug design. If they know the 3D structure of the target, like having its detailed blueprint, they can computationally predict which virtual compounds might fit well, like finding the right key shape for a specific lock.
Okay.
Then there's ligand-based design. That looks at compounds already known to work on similar targets and tries to find other molecules with similar features. These computer methods can drastically cut down the number of compounds they need to actually make and test in the lab.
So they narrow it down from potentially millions to a smaller set of hits. Are these ready for human testing then?
Oh, not by a long shot, no. These initial hits, they're usually pretty rough, far from ideal drug candidates.
How so?
Well, they might not be very potent, meaning you'd need a huge dose. Or they might hit other molecules in the body too, causing side effects. Or maybe they just don't get absorbed well or get broken down too quickly.
Right.
That's where the next stage, lead optimization, comes in. It's this iterative process, refining those hits, enhancing the good properties, minimizing the bad ones, basically turning a rough stone into a more polished gem.
And what exactly are they trying to improve during lead optimization? What makes a polished gem in this context?
Think of it like fine-tuning an engine. Key goals are improve potency, so a lower dose works.
Makes sense.
Increase selectivity, make sure it hits the disease target specifically and not lots of other healthy stuff.
Fewer side effects.
Exactly. Then optimize pharmacokinetics, that's ADME. Absorption, distribution, metabolism, excretion, basically how the drug gets in, travels around, gets processed, and leaves the body. It needs to reach the target and stick around long enough.
Okay, ADME.
Got it.
And of course, constantly reducing potential toxicity.
How do they do that? Just keep tweaking the molecule. Pretty much. Medicinal chemists make small changes to the chemical structure of the hits, creating analogs. Then they test these new versions rigorously, see how those structural changes affect biological activity. This generates what we call structure-activity relationship data, or SAR, understanding exactly how the structure relates to function.
This SAR.
And they also use computational models, quantitative structure-activity relationship, QSR, to help predict properties of new variations before they even make them.
So it's a cycle. Design, make, test, learn, repeat.
Precisely. A continuous cycle. Experimental results feed the computer models, and the models suggest new experiments. The ultimate aim is to identify one or more lead candidates.
Lead candidates.
Yeah, molecules with a really good, balanced profile. Good activity, behaves well in the body, decent safety margin, making them suitable to finally move into the next big phase, preclinical research.
Preclinical research. Okay, now that does sound like we're getting closer to human trials. What happens here?
You're right. This is really about laying the foundation for testing in people. Preclinical research is all about gathering tons of safety and biological activity data before you even think about giving the drug to a human.
So lots more lab work. And animals.
Exactly. It involves a significant amount of work, both in vitro, that's using cells, tissues, in the lab.
Okay.
Which helps researchers really pick apart the drug's mechanism of action. And in vivo studies using animal models designed to mimic specific human diseases.
Why the animal models?
They're crucial for seeing how the drug behaves in a whole living system, how it gets absorbed, where it goes in the body, what effects it has systemically, any unexpected consequences.
And I guess this is where they really dig into how the body handles the drug, that ADME stuff.
Absolutely.
Yeah.
This is deep into pharmacokinetics, PK. What the body does to the drug. Absorption, distribution, metabolism, excretion, how does it journey through the body?
Right.
And at the same time, they study pharmacodynamics, PD, what the drug does to the body, its actual effects, how strong they are at different doses.
PK and PD.
Yeah, and they often use PK-PD modeling, linking the two, trying to use the animal data to predict how the drug might behave in humans and figure out a safe starting dose for those first human trials.
Safety seems like the absolute key word here.
Yeah.
What kind of tests do they run to see if these candidates might be harmful?
Oh, it's critical. Toxicology and safety pharmacology studies are huge here, designed specifically to find potential hazards and establish that safe starting dose for humans.
What kinds of hazards?
All sorts. They run single-dose tests for immediate effects, repeated dose tests for longer-term exposure issues. Safety pharmacology looks at vital systems, heart, lungs, brain. Is the drug affecting those?
Okay.
Then there's genotoxicity. Does it damage DNA? Carcinogenicity, could it cause cancer, especially for drugs meant for long-term use?
Wow, thorough.
And reproductive and developmental tox studies, any harm to fertility or a fetus, immunotoxicity effects on the immune system. It's a very comprehensive battery of tests.
I read about something called the NOAEL, no observed adverse effect level. What does that mean?
Ah, yes, the NOEL. That's the highest dose tested in animal studies where they saw no significant harmful effects.
Correct.
And that NOAEL value is really key. It's used in the calculations to determine a safe starting dose for the very first human clinical trials. It's a bridge from animal safety data to human safety.
Are there like strict rules for how these preclinical studies have to be done?
Oh, absolutely. Especially for these pivotal safety studies. They must follow rigorous guidelines called Good Laboratory Practice, or GLP.
GLP.
Yep. GLP covers everything: qualifications of the staff, the equipment used, how data is recorded, analyzed, reported. It ensures the quality and integrity of the data going to regulators.
Right.
And of course, there are major ethical considerations around animal use. The 3Rs principle is key: replacement, reduction, refinement, trying to minimize animal use and suffering.
That's important.
Definitely. And it's worth noting, animal models aren't perfect replicas of humans. There's a big push to develop better, more predictive systems.
So while all this biology and safety testing is going on, are they also figuring out how to actually make the drug if it gets approved, like on a big scale?
That's a really sharp question. Yes, absolutely. In parallel, there's a whole critical stream of work called chemistry, manufacturing, and controls, CMC.
CMC.
Essential stuff. Developing a reliable, scalable process to make the active drug substance. Figuring out the formulation, tablet, capsule, injection, ensuring it's stable over time. Setting up strict quality control tests for purity and potency.
Because you could have a great drug, but if you can't make it reliably.
Exactly. If you can't manufacture it consistently and to a high standard, it's never going to reach patients. CMC is crucial.
Okay, so after potentially years of discovery, optimization, preclinical testing, CMC development, if they think they have a winner safe enough, potentially effective, what's the next massive hurdle?
Getting permission to test it in humans, that means compiling all that preclinical data, plus detailed plans for the first clinical trials, into an investigational new drug application, an IND in the U.S.
There's CTA in Europe, right?
Correct. A clinical trial application, or CTA, in the EU and other regions. This application is basically the culmination of all that foundational work. It's the gateway to clinical development.
An IND application. Sounds like a beast of a document.
It really is extremely substantial. Contains all the chemistry and manufacturing details, all the pharmacology and toxicology findings from animals, detailed protocols for the proposed human trials, exactly how they plan to study it in people.
Do they just submit it and cross their fingers?
Well, often they talk to the FDA even before submitting. There are pre-IND meetings and even earlier interact meetings for very novel therapies to get feedback on their plans and data.
That makes sense. Get guidance early.
Definitely. It can really help smooth the process. Then, once the IND is submitted, the FDA reviews it thoroughly, mainly focusing on whether it's reasonably safe to start those first human studies.
And how long does that preclinical phase usually take up to the IND?
It varies a lot, but typically you're looking at maybe 4 to 7 years before they get that green light to move into clinical trials.
Okay, so the IND is approved. Finally, we get to testing in humans. This feels like the part everyone hears the most about. Can you walk us through the different phases? Phase 1, 2, 3.
Absolutely. Clinical development, this is usually the longest and definitely the most expensive part of the whole journey. It's where the investigational drug is systematically tested in human volunteers and patients.
To check safety, if it works, the dose.
Exactly. Safety, effectiveness, optimal dosage. And it's typically structured in sequential phases, each building on what was learned before.
Right. Phase 1, 2, 3. But I've also heard of a phase 0. What's that about? Is it common?
Phase 0 is relatively new and it's optional, sometimes called exploratory IND or microdosing studies.
Microdosing.
Yeah, they give tiny, tiny doses, subtherapeutic, not expected to actually treat anything, to a very small number of people, maybe just 10 or 15.
Why?
The main goal is to get really early human PK/PD data, see how the drug is absorbed, processed, does it hit the target in the body, or maybe quickly screen several candidates to pick the best one to move forward.
Like a quick preliminary check in humans.
Kind of, and because the doses are so low, the regulatory hurdles are a bit lower, so it can potentially speed things up a little.
Okay, so phase 0 is like a tiny first glimpse. Then comes phase 1. What's the main focus there?
Phase 1 is all about safety and tolerability. This is the first time the drug is given at doses intended to have a biological effect, though they start very low and carefully increase.
So finding a safe dose range.
Precisely. Determining a safe dosage range, identifying side effects, and figuring out the maximum tolerated dose, or MTD.
MTD, the highest dose without unacceptable side effects.
Exactly.
And who normally takes part in phase 1?
Usually it's a small group, maybe 20 to 100 of healthy volunteers.
Healthy people, not patients.
Typically, yes. But for some drugs, like maybe cancer drugs or others expected to have significant side effects even at low doses, phase 1 might be done in patients who actually have the disease.
Because it wouldn't be ethical to give it to healthy people.
Right, if there's significant risk and no potential benefit for them.
I've heard terms like single ascending dose and multiple ascending dose. What are those?
Good question. PsyD studies single ascending dose. Sometimes phase 1A involve groups of participants getting just one dose. If it's safe, the next group gets a higher dose, and so on.
Until they hit problems?
Until they reach the MTD or unacceptable side effects. MAD studies multiple ascending dose. Phase 1B give participants repeated doses over days or weeks.
Why repeated doses?
To see how the drug builds up in the body, check safety with longer exposure, and look at the PK/PD over time. They monitor everyone incredibly closely for any adverse events.
How do they decide how much to increase the dose each time?
They use specific dose escalation schemes. The classic is the 3+3 design, common in oncology. Treat 3 patients. If no serious side effects, escalate. If one has issues, add 3 more at that level. If 2 or more have problems, that's usually the MTD.
Okay.
But more sophisticated adaptive designs, like the continual reassessment method, CRM, are increasingly used now. They adjust doses based on ongoing results to find the MTD more efficiently.
And how many drugs make it past phase 1?
Roughly 70% successfully move to phase 2. Failure is usually due to unexpected toxicity or bad pharmacokinetics. The drug just doesn't behave well in the human body.
Okay, so 70% make it to phase 2. The focus shifts now, right?
Big shift. Phase 2 is primarily about efficacy. Does the drug actually work? Does it show the intended therapeutic effect in patients with the disease? This is the initial proof of concept stage.
Proof of concept. And they're still watching safety.
Absolutely. Safety monitoring continues, but now in a larger group of actual patients. And they're also trying to refine the dosage, find the best dose or doses for the big phase 3 trials.
Are phase 2 studies designed differently? More patients?
Yes, typically more patients, maybe dozens up to around 300. And crucially, these are patients with the target disease.
How do they measure if it's working?
They often use randomized controlled designs. Some patients get the drug, others get a placebo or an existing treatment. Frequently, yes. Double-blinded is ideal. Neither patients nor researchers know who gets what to minimize bias. They look at clinical endpoint, real measures of patient improvement, like symptom reduction, or sometimes surrogate endpoints markers like tumor shrinkage that are thought to predict benefit.
Surrogate endpoints, like a shortcut measure.
Kind of. They also focus on finding the right dose, not just the highest tolerated, but the optimal biological dose, OBD, as the FDA emphasizes with Project Optimus, the dose giving the best balance of benefit and risk.
Are these trials adaptive too?
They can be. Designs like Simon's 2-stage allow them to stop early if the drug clearly isn't working, saving time and resources.
And success rates here, you said phase 1 was 70%.
Phase 2 is tougher. Historically, the success rate moving from phase 2 to phase 3 has been lower, maybe around 33% or even less.
Wow, that's a big drop. Why?
Often, it's simply insufficient efficacy. The drug just doesn't work well enough in actual patients. Or sometimes unexpected safety issues pop up in the disease population. It's a really critical make-or-break stage. Robust phase 2 design is vital.
Okay, so if a drug does show good signs in phase 2, it graduates to phase 3, the big ones, right?
Exactly. Phase 3 trials are the large-scale pivotal studies. They aim to definitively confirm efficacy in a broad patient population and gather much more comprehensive safety data.
Looking for rarer side effects.
Precisely. Things that might only show up when hundreds or thousands of people take the drug or take it for longer periods.
How many people are usually in phase 3?
Hundreds, often thousands. And they're usually multi-center, often global, to get results applicable to diverse patients and settings.
And the main goals?
Get statistically significant proof the drug works better than the control, placebo, or standard care. Build a very detailed safety profile, see how it works in different patient subgroups.
Still randomized, controlled, double-blind?
Almost always, yes.
Yeah.
These are the gold standard for minimizing bias. The data from phase 3 is what primarily supports the application for marketing approval.
What's the success rate for phase 3?
Yeah.
Getting to that submission stage.
Sure. Estimates vary, but maybe around 25–30% of drugs entering phase 3 make it to an FDA submission. The rate for simply completing phase 3 might be higher, but completion doesn't guarantee approvability.
Still pretty tough odds. Late-stage failures must be devastating.
Incredibly costly and disappointing, yes.
Yeah.
It highlights the challenge. And these trials are getting even more complex now with adaptive designs using biomarkers to select patients.
Yeah.
It's always evolving.
Okay. So phase 3 is successful. Positive results. What's the very next step to get the drug to patients?
That success unlocks the door to regulatory review. The company prepares a massive application for marketing authorization.
The NDA or BLA in the U.S.
Exactly. New drug application, NDA, for small molecules, biologics license application, BLA, for things like vaccines or antibodies, or the MAA marketing authorization application in Europe.
And these applications must be enormous, right? What's in them?
Absolutely huge. They contain basically everything. All the discovery data, all the preclinical results, all the clinical trial data from phase 1, 2, and 3, detailed manufacturing info, quality control, everything.
The whole story.
The whole story. Meticulously documented. The goal is to prove to regulators that the drug is safe, effective for its intended use, and can be made consistently to high standards. Compiling it takes months, huge teams.
I've heard of the CTD format, Common Technical Document.
Ah, yes, the CTD. That's a standardized format for organizing these massive submissions. It's accepted by the US, Europe, Japan, many others.
Makes it easier for everyone.
Definitely. Easier for companies preparing submissions for multiple regions, and easier for the agencies to review efficiently. It has 5 modules: admin info, summaries, quality CMC data.
CMC again. Sounds important.
Module 3, quality CMC, is critical. All the manufacturing details are there. Then Module 4 is non-clinical reports, animal lab studies, and Module 5 is all the clinical study reports.
Okay, so the application NDA, BLA, MAA is submitted. What happens then? How does the FDA or EMA review it?
It's a very rigorous process at both agencies. Let's take the FDA. First, an initial check is the application complete enough to review. Then for a standard review, they aim for a decision in about 10 months from filing.
Only 10 months. Seems fast for all that data.
Well, it's intense, but they can grant priority review for important drugs, shortening it to about 6 months.
How do they review it?
They have multidisciplinary teams, doctors, statisticians, chemists, pharmacologists who scrutinize everything. For complex or novel drugs, they might hold an advisory committee meeting.
External experts.
Exactly. Independent experts give input. They also conduct pre-approval inspections, or PAIs, of the manufacturing plants. Super important.
Checking the CMC in person.
Right. And they might inspect clinical trial sites, too. Then there are negotiations over the drug's label, the prescribing information,
And the outcome.
approval, or a complete response letter, CRL, meaning issues need fixing before approval, or rarely rejection.
And the EMA process in Europe. Similar.
Similar rigor, different structure. Many innovative drugs go through a centralized procedure for EU-wide approval. The scientific review is done by the CHMP Committee for Medicinal Products for Human Use. That takes up to 210 active review days.
CHMP gives an opinion.
Yes, a positive or negative scientific opinion. The final decision is then made by the European Commission, usually within about 67 days, and the drug gets listed in the community register.
It sounds incredibly thorough. But what about speeding things up for really critical drugs? Are there ways to do that?
Yes, definitely. Both FDA and EMA have expedited pathways for serious conditions with unmet needs.
Like what?
FDA has fast track, breakthrough therapy designation, accelerated approval, priority review. EMA has PRIME, accelerated assessment, conditional marketing authorization.
What do they do?
They offer things like more frequent meetings, potential for rolling review, submitting sections as they're ready, shorter review timelines. Accelerated approval is interesting approval based on a surrogate endpoint.
The marker predicting benefit.
Right, but with a commitment to do post-marketing studies to confirm the actual clinical benefit later.
Conditional approval in Europe sounds similar.
Somewhat, yes. Earlier access based on less data initially, with requirements for more data later. But these pathways, while faster, often mean more intense scrutiny, especially on the manufacturing side, the CMC.
Okay, so finally, approval granted. The drug is on the market. That's the end of the line, right? Start selling.
Ha! You'd think so, but no. Getting approval is huge, but it's definitely not the end. Ongoing monitoring is crucial once the drug is used by thousands, maybe millions of diverse patients in the real world.
So more monitoring.
Absolutely. Post-market surveillance. This includes phase 4 studies and the really important field of pharmacovigilance.
Pharmacovigilance sounds serious. What is it?
It's basically the science of detecting, assessing, understanding, and preventing side effects, adverse drug reactions, ADRs, or any other drug-related problems after approval.
Because things might show up later.
Exactly. Rare side effects, issues with long-term use, problems in specific groups, things you might not see even in large phase 3 trials.
How do they track this?
A key tool is spontaneous reporting systems. Doctors, pharmacists, patients, companies report suspected ADRs to regulators. FDA has FAIRs, EMA has vigilance.
And regulators watch these reports.
Closely, they look for patterns or safety signals. If concerns arise, they can take action, update the label with warnings, restrict use, or in rare, serious cases, withdraw the drug.
And you mentioned phase 4 studies, too. How do they fit in?
Phase 4 studies are clinical trials done after approval. Sometimes regulators require them, especially for accelerated approvals, to confirm benefit or long-term safety.
Or companies do them voluntarily.
Yes, often. To get more long-term data, compare their drug to others, study it in new populations, or even explore new uses for it. These can be large, long-running studies giving valuable real-world insights.
It seems like we have so much more data available now once a drug is out there. What about things like real-world evidence and AI? Are they changing this post-market phase?
Hugely. You're absolutely right. Real-world data, RWD, from electronic health records, insurance claims, registries, even wearables.
Data from actual patient care.
Exactly, and analyzing that data generates real-world evidence, RWE. It's becoming incredibly important for monitoring safety and effectiveness in routine practice.
And AI helps analyze all that data.
Massively. AI and machine learning can sift through vast amounts of RWD much faster than humans. They can help detect safety signals earlier, predict risks, automate processing of ADR reports, even analyze doctor's notes using natural language processing.
So it's becoming more proactive, less reactive.
That's the goal, moving towards a more proactive, predictive system, though there are still big challenges with RWD data quality, completeness, standardization that need work to really unlock its full potential with AI.
Wow. This whole process from discovery to post-market, it's just incredibly complex, so many different factors pulling together.
You really hit the nail on the head. It's an extraordinary interplay of science, logistics, finance, regulation, ethics, all deeply interconnected.
Let's talk numbers for a second. We hear mind-boggling figures. What's a realistic snapshot of the timelines, costs, and, frankly, the chances of success?
The numbers are striking. Timeline. Average, maybe 10, 15 years from discovery to patient. Sometimes much longer for really novel stuff like gene therapy.
A decade or more.
Easily. Costs. The often cited figure, including failures, is around $2.6 billion capitalist costs per approved drug. Global R&D spending is over $300 billion annually.
Staggering amounts.
And success rates? Well, they're sobering. Only a tiny fraction of initial ideas make it. We talked about the phase transitions, maybe 70% past phase 1, only 33% or so past phase 2, maybe 25-30% past phase 3 to get submitted.
So the overall chance is tiny.
Very small. Recent data suggests the phase 1 to approval rate might be down around 6.7%. It really highlights the risk. And the huge incentive to fail fast, fail cheap, weed out the losers early before those massive phase 3 costs hit.
Beyond time and money, what are some of the recurring headaches, the common scientific, logistical, ethical challenges?
Scientifically, finding truly novel targets is hard. Translating promising lab or animal results into humans is a huge hurdle, the valley of death. Patient variability is another big one. Why does a drug work great for one person and not another?
Right.
Logistically, running huge global trials is incredibly complex. Recruiting enough patients, keeping them in the study, managing drug supply chains, ensuring data quality across many sites. Protocols get more and more complicated too.
Sounds like a nightmare to manage.
It can be, ethically. Participant safety is always number one. Informed consent, using placebos appropriately, ensuring fair access to trials, data transparency, responsible animal use. These are constant, vital considerations. All these challenges contribute to why drug R&D productivity can sometimes seem low despite massive investment.
This has been just an incredibly eye-opening deep dive. It's so clear how rigorous, how challenging, how long this journey is. Just immense dedication involved.
It really is.
As we wrap up, what are the biggest takeaways you think listeners should hold on to? And what's on the horizon? Where is this field heading?
I think a key takeaway is just the sheer rigor and complexity. From that first idea to ongoing monitoring years later, it's all driven by this commitment to safety and efficacy. We saw the stages, the collaboration needed scientists, doctors, regulators, patients.
It's a huge ecosystem.
Truly. And looking ahead, it's exciting. We're seeing rapid advances in new types of therapies, gene, cell therapies, big pushes towards personalized precision medicine.
Tailoring treatment.
Exactly. AI and real-world evidence are making development smarter, faster. Decentralized trials are reaching more people. There's more focus on patient centricity, listening to patient needs throughout. And regulators are evolving too, trying to keep pace.
Things are getting better, more efficient.
There's definitely a convergence of these trends, creating a more integrated, data-driven, adaptive system. But big challenges remain. The innovation gap, still many diseases without good treatments, those escalating costs, the low success rates overall, harmonizing regulations globally, and the ethical questions raised by new technologies.
So it's still evolving.
Constantly. The future really hinges on adapting quickly, on regulators fostering innovation while ensuring safety, and honestly, on even more collaboration between everyone involved to get needed treatments to patients worldwide.
Which leads to a final thought for you listening. Having heard this overview, what part of this whole intricate process surprised you the most? Or, maybe, what do you think is the single biggest hurdle we still face in getting new medicines to the people who need them? Something to mull over.